Okay, so the second talk is was supposed to be a little bit more of a research talk. So I thought I'd give you an idea about fluorescence correlation spectroscopy as a tool, as we use it at least in developmental biology in zebrafish. And that is a story of Wnt3, a molecule we've been working on for quite some time. So that work has started together with Vladimir Kors and Kathleen Tay. Kathleen Tay is now in my group. And the PhD students working on that was Gangyu Sun, Shivan An, and Saptasran Vedapatian. All of them have actually already finished their thesis. Sapta just left and just published the most recent paper on it. Okay, so let's start. So if you look in biology, actually, among the top 5 to 10 questions, unsolved questions in biology, you always find developmental biology. And the question there's always, you start with one cell, and then after 30 hours or so, you get actually something that looks already somehow something like an organism. How does it happen, right? If you would have just one cell and just continuously dividing, there would be no structure. You'd have just a large blob. Obviously, that's not what happens. And so we have a couple of molecules that control this, and they break the symmetry. Right? So while you start out with something which is very symmetric, you end up with something which is relatively complex. And there are a number of different morphogens or signaling molecules that control this, and one of them is actually Wnt3. And we measure on all of them, but in particular Wnt3 for this talk. Now, what is the problem? The problem is that if we measure and this is an example of nodal factors, another morphogen, if we measure the gradient of these molecules within the fish, and it's these gradients, right? If cells sit at different concentration thresholds, they will develop differently. So if you measure these gradients, they are somewhere like, I don't know, 20 to 40 micrometer long. If you measure the diffusion coefficients of nodal factors in our case are about 60 micrometers squared per second. And that's a problem. If you have 60 micrometers squared per second diffusion coefficient, there is no way that you get that you can keep a gradient of 20 to 40 micrometer for a very long time. It's just going to equal out, and your embryo is basically filled with your morphogen. If you want to have a gradient, as we measure, you need actually diffusion coefficient, which is about 2 orders of magnitude lower. So how can that be, right? That we measure so fast diffusion coefficient, but what we actually need is actually much lower diffusion coefficients. Where the issue? And that is what we try to solve for Wnt3. So Wnt traffic, so Wnt's are in general secreted hydrophobic proteins. They're involved in tissue patterning, polarity induction, morphogenesis in a developing embryo. And in particular, Wnt3 is that's marked here, right, with a one or multiple lipid tails. We'll take a look at that. And once these Wnt's reach their target tissue, they bind to their receptors and then start signaling, which is typically beta-catenin signaling, but can be as well independent of beta-catenin signaling. So what we try to answer is, how is Wnt3 lipidated? Are there 1, 2, 3, 4, 5 tails, whatever? How is Wnt3 transported? So if Wnt3 is hydrophobic, actually, can it actually diffuse in these very, very narrow spaces? And as you'll see in a moment, these spaces in the zebrafish are very narrow, 40 nanometers or so. So they are too small for us to actually directly image. And finally, how does Wnt3 interact with this receptor and start signaling? So to start this and to take a look at Wnt3, we have transgenic animals. Fish we made. They are driven by a 4 kilobase Wnt3 promoter, which leads to expression patterns which are very, very similar to the wild type Wnt3. And we drive Wnt3 EGFP, so Wnt3 is labeled as EGFP, and the transgenic line we have actually multiple families. We work with one family, and they were created by Toll2 transposon transgenics. So here for your orientation, so that's a zebrafish with eyes. We have here the midbrain hindbrain boundary. That's very important for us. right? Here's a hindbrain, and here we have the roof plate. So we have multiple lines, actually. We have, as I just said, we have the Wnt3 EGFP line. We have another line that expresses PMTmApple. PMT is a molecule, is actually a small peptide. It's labeled with a fluorescent protein called mApple. It's a red fluorescent protein. And PMT is just carries two lipidation sites, a methylation, palmitoylation site. So it actually sits on the inner membrane of your cells. So the first thing we can do now is we can take a look is Wnt3 at all transported in the animal? How do we do that? We actually cross these two lines, and both lines are running on the same promoter. So PMTmApple, as it's in the inside of the membrane, cannot move through the fish at all. It has to stay at the cells where it is expressed. But Wnt3 EGFP, if we are right, then it will find it at the cells it's expressed, but it will be as well secreted, and it will actually reach other cells. So if you think about that, so all cells that carry Wnt3 EGFP and PMTmApple are the cells that express Wnt3, and all the cells which contain only Wnt3 but no PMTmApple are the target cells. That's where Wnt3 moves to. And that's what you actually see here. So that's the Wnt3 EGFP. We have here 24 and 48 hours. So what you see here is a cerebellum, the midbrain hindbrain boundary. This here is an optic tectum. And here that's a fourth brain ventricle. That's a large fluid-filled ventricle. So, and here you see the PMTmApple. Right, so you see that clearly that Wnt3 distributes over a much larger region than PMTmApple, right? Whether it's at 24 or 48 hours, clearly indicating that Wnt3 actually diffuses away from the origin where it was actually originally produced. Okay. So that led us now to well, this still doesn't answer really how it is actually moving through your fish. It could still be that it's somehow handed on over liposomes or handed over from cell to cell, or it could be diffusion. So we try to see whether it is diffusion. So what we've done, we have taken here 3 different zebrafish. One is Wnt3 EGFP, our normal fish line. One with Lyn EGFP. Now, Lyn EGFP is as well, again, a membrane-bound non-functional Lyn kinase, actually, with the EGFP. So we see actually the EGFP only in the membranes, and we get as well a secreted EGFP, which is doing exactly the opposite. It is actually gets all secreted. So you see very nicely that secreted EGFP we can find in the brain ventricle. So we have done now, we've gone to these different cell lines, and we have started here from the cerebellum and made just a line scan, an intensity scan in this direction. And we've done this for all 3 lines. You see on this one, the secreted EGFP line, we have an additional red marker to just show where the membranes are, where the cell membranes are. So what we find is that secreted EGFP in blue very clearly is, of course, found in the brain ventricle. We normalized this actually too. If you take a look at Lyn EGFP, a molecule that should be only on the membrane, you see actually very nicely, we see here the peak on the membrane, but then outside in the brain ventricle, there is no Lyn EGFP, right? There is no Lyn EGFP because Lyn EGFP is not secreted, it's bound to the membrane. But if you look at Wnt3, the red one, you see that we find Wnt3 on the membrane, right? Very clear, you see that here already, but we find it as well in the brain ventricle. And that indicates already that Wnt3 is secreted and can diffuse. And here are the correlation functions, right? So wild type, so autofluorescence, you see little correlation function. Lyn EGFP is the same thing. You don't see a correlation function in the brain ventricle, but you see correlation functions of secreted EGFP and as well Wnt3 EGFP. And Wnt3 EGFP is a bit wider. Why? Because Wnt3 EGFP is, of course, as well larger than EGFP alone. So it's expected to have a wider correlation function. The problem for us is now we don't really know what models to use. Is this a 1-particle model, a 2-particle model? And since we have a certain amount of noise on there, can we make a difference between these 2 models? And I think I will not go through this in too much detail, but what we've done is if you take a look at the places we measure, right? You have one cell here, one cell here. If you measure on here, we see cytoplasm on both sides, and we see the extracellular space between, and we see the two membranes. So it could be a 2-particle, 3-particle model. It could be a 1-particle model. We don't know, depending on where the molecules actually are. So we use something which is called Bayesian fitting, and I'm not going to go through this right now. The important thing is Bayes fitting allows us to determine the probability of a particle of a diffusion coefficient model or FCS model to be correct, independence on the noise you measure. So if you have large noise, then your probabilities for the different models going to change, right? So because then you can't use more parameters, right? So Bayes actually punishes for you for having large noise and having too many parameters. Okay, so what we did now, we mount our zebrafish and make our measurements in the brain ventricle, and then we focus on the membrane, we measure, and we try to figure out what is the right diffusion coefficient model. And that's what I want to show you here. So what I show you here is the different models we've tested. So 1P is a 1-particle model. 1P, 1T is 1 particle with 1 triplet, so with 1 photophysics contribution, right? A 2P would be a 2-particle model. That means we have 2 different diffusing particles. 2P, 1T would be 2 particles and 1 photophysics component, and 1P, 2T would mean we have 1 particle diffusing, but it has 2 different kind of photophysics going on, right? So, and on the y-axis, you see the probability for each model. So let's start with secreted EGFP. That's an EGFP, the blue ones, and what you see here, and the blue and black ones, sorry. And what you see here is the highest probability we get clearly for 1 particle and 1 triplet, because we have 1 fluorescent protein, secreted EGFP or EGFP, right? And we see actually from EGFP always a little bit of photophysics, of some blinking of EGFP, right? That's why the 1 particle, 1 triplet model has, in this case, the highest probability. So that would be the model we select. If you go for Lyn EGFP, we get we get the highest probability for a 2-particle model and 1 triplet. That means we see 1 triplet because we see the triplet of the EGFP, but we see the EGFP diffusing with 2 different diffusion coefficients. And for Lyn, I'll show you in a moment, this is probably because we see something on the membrane, and we presumably see something intracellularly. For Wnt3 EGFP, same thing. The highest probability is actually clearly the 2-particle, 1 triplet model, which means we see at least 2 different diffusion coefficients for Wnt3 and a triplet. And here, that's the typical correlation function for Wnt3 we see. It's just to show you that we can actually fit them actually quite nicely, right? And we can extract the different numbers. So the green one is the correlation function, the experimental one. And then we see 1 diffusion coefficient here with a time of 29 milliseconds, a much faster diffusion coefficient, less than a millisecond, right? And then we see as well actually some triplet here in the beginning. And of course, we can determine as well the fractions of all of these molecules. Okay, so let's compare Wnt3 and Lyn. That's a D1. The D1 is a fast diffusion coefficient, right? It's the order of tens of micrometers squared per second. And you see that we see Lyn EGFP a little bit faster, but well. D2 is a much smaller diffusion coefficient, right? At least 10 times smaller than what we see here, right? And you see here a diffusion coefficient on the order of micrometers squared per second, also for Wnt3 EGFP and for Lyn EGFP on that order. So interestingly now is we see if we compare Wnt3 EGFP and Lyn EGFP and we look at the second fraction, the fraction of the slow-moving particles, then we see always a higher fraction of slow-moving particles for Lyn EGFP. Right? Here you see actually all the measurements we've done, you see the plots, and you see very nicely that Lyn EGFP has always a larger fraction of slow-moving particles compared to Wnt3 EGFP. That means more of Wnt3 EGFP is moving fast. How can we explain that? Our explanation for that is Lyn EGFP is sitting on the membrane, or we see some of it in the cytosol because it has, of course, to be transported to the membrane, and right. But we don't see anything in the extracellular space because Lyn EGFP is not secreted. Wnt3 EGFP, by contrast, we see, of course, the Wnt3 on the membrane. That's the slow diffusion coefficient. But the fast diffusion coefficient, we see two parts. We see actually the cytosolic part here and here in these two cells, but we see an extracellular part. And that is a reason why for Wnt3 EGFP, we think we see always a higher fraction so a lower fraction of slow-moving particles, a higher fraction of fast-moving particles, because we get Wnt3 moving in the extracellular space. So it's the second evidence for us that we get actually Wnt3 secretion and diffusion in these extracellular spaces. Now, if Wnt3 moves in the extracellular space, the question then, how does it do that? Because it has actually lipidation. So that means it's hydrophobic. So it's not easy to be in water. So what the typical assumption is that Wnt3 actually interacts with heparan sulfates that allows it to hop on and to hide its hydrophobic tail. So if that is true, then if we remove this heparan sulfate proteoglycans, HSPG, with a molecule called heparinase, then we should see a change of diffusion of Wnt3. So let me just show you the measurements. So Wnt3 EGFP has a diffusion coefficient, the fast diffusion coefficient on the order of 25 micrometers squared per second. The moment we disrupt the heparan, the HSPG, the diffusion coefficient goes up. If you do this for Lyn EGFP, there's no change. Why? Again, Lyn EGFP is not in the extracellular space. If you do that for secreted EGFP, nothing changes. Why does nothing change? Because secreted EGFP doesn't interact with HSPG. But for Wnt3 EGFP, clearly we see a change of diffusion coefficient. That means A, Wnt3 EGFP interacts with HSPG, and B, it must be in the extracellular space because that's the only space where HSPG is. So clearly showing again that we get a Wnt3 secreted. So we did now a couple of FRAP experiments as well, because that will help us to figure out what actually happens in the movement outside. Because remember, the diffusion coefficient we measure is now 25 micrometers squared per second. But if you want to build any kind of gradient, we have to be much smaller. So if we do a FRAP in a PMTmApple system, we don't get any recovery. Why? Because FRAP is intracellular, so there's no recovery from other cells once you bleach the PMT away. And that's, of course, expected. What happens if we do FRAP for Wnt3 EGFP? Here clearly, so that's pre-bleach, that is bleached, and that is after recovery, right? So we get recovery, and the recovery, we have actually two different components, a fast component and a somewhat slower component, where the slower component might be related to protein expression. And we have about 35% of the molecules that are mobile. Okay. So if we measure the diffusion coefficient with FRAP, we get 0.5 micrometers squared per second. Okay, after everything we've done, we're back to step one, right? I mean, again, we have a factor 50 between FRAP and FCS. We have to explain somehow. Where does factor 50 come from? Exactly the same as I told you earlier, right? So the first thing is we have to take a look at is, does FRAP and FCS even give you the same diffusion coefficients? Because FRAP measures over a large area and FCS over a very small area. So what we've done, we use total internal reflection microscopy. So they illuminate just the 100 nanometers or so above a cover slide, right, with an evanescent wave. And then, as I told you earlier, if you have actually a camera, you can actually get every single pixel a correlation function. So what we have done now, we have done that on a cell. And while we measure FCS, we bleach. So we do actually a FRAP experiment while we're doing FCS. So, and you see here the number of particle map, right? This is pre-bleach. Here we bleached an area on the cell membrane. And here we see the recovery. I should have said that this is actually GFP, GPI on the membrane in CHO cells, right? So you see clearly nicely where we bleach. If you look at the diffusion coefficient, it never changes. It stays exactly the same. So the first thing is FRAP itself does not influence our FCS measurements, which is good, right? And then we can, if we can measure the diffusion coefficients from one or the other, we get about the same values. Now, we measure this over a whole number of different different cells, and we did this as well for different simulations. And what comes out of the whole thing is like, I do this shortly, is actually that FCS and FRAP give you more or less the same diffusion coefficients, so that they're fully consistent. It is only that for FRAP, because FRAP has more of a problem with finding actually the right fit models, has a much wider variability of the diffusion coefficients it gives you, so it has a larger error. And that's what you see here. And that's pretty much as well what you see here. So the error in this direction for FRAP is higher than what you see in FCS. Interestingly, we had one cell which was a complete outlier, but it was an outlier for both FCS and FRAP. So that's that there was something different with the cell we don't know, but the good thing for that is it's an indicator, again, that FRAP and FCS are fully consistent. They don't give you different values. They should give you exactly the same value if they measure the same thing. Okay, so that doesn't help us at all, because that means we still have to explain this factor 50 of difference, because which we see between FCS and FRAP. So let's take a look at FRAP of secreted EGFP. That's EGFP that just diffuses outside, right? So if I take a look at the recovery, I can more or less fit this as well, or I estimate at least the diffusion coefficients. The fast component has a recovery time of 1 minute, high mobile fraction. And what we get now is with FRAP, we get 30 micrometers squared per second, while with FCS, we had, for EGFP, about 60 micrometers squared per second. So we get a factor of 4 difference between FRAP and FCS. And the major difference between the two is that actually FRAP measures over a larger area, while FCS measures basically just at one single space between the cells in the extracellular space. So what that means is actually the recovery very likely is slowed down because of the geometry of your system. That's what we call tortuosity. So a molecule cannot just move straight from here to here, but it has to actually go around a complicated path before it can arrive here. That slows it down. And this factor, this geometry-based factor, this tortuosity, is about a factor 4. It's not our factor 50, but at least a factor 4 we have explained. So the next thing we did, we took Wnt3 EGFP after we disrupted HSPG. So if we do this and fit this data again, we get actually diffusion coefficients of FRAP of about 0.5 micrometers squared per second before we disrupted HSPG. After HSPG, we get about 2.6 micrometers squared per second. So what that means is we have about a factor 5 of slowdown explained by Wnt3 actually binding and interacting with HSPG. So you can imagine that Wnt3 is not only that it has to go around the cells, but while it moves around the cells, it constantly interacts with HSPG, and because of that, slowed down. So we have now a factor 4 and a factor 5 together. That gives about a factor 20. Right? So we have now explained actually almost a factor 20 already. But is that all? Before we can answer that, we have to use fluorescence cross-correlation spectroscopy, because we're now interested whether there are other binding partners, which of course would be, in this case, the receptor. Cross-correlation spectroscopy I already explained to you, right? And I've already shown you that actually you get from the two autocorrelation to the cross-correlation function, can estimate or can calculate how many molecules interact, and you can get a KD from there. So we looked at Wnt3 and frizzled one. Frizzled one is one of the major targets of Wnt3. So it's one of the large family of frizzled receptors that interact with Wnt. And we did this with FCCS. And the interesting thing is So this is a positive control. We have a PMTmApple EGFP. We get the autocorrelations and the cross correlation. That's the best case we can get. We can't get better than that. This is a negative control that is Wnt3 EGFP and PMTmApple. So the two have no reason to interact. Right? There's two completely different molecules, and that's exactly what you see. You get absolutely no cross correlation function. I should mention here that to get such a nice negative control, what we do is we alternate our lasers. We switch the green on and the red is off, and then we do the other way around. In this way, you can avoid crosstalk and you get actually very nice negative correlation functions. So that's a positive and negative control. And if we measure Wnt3 together with frizzled mApple, you see actually clearly positive correlation function. And if we calculate our KD from there, we can do our scatter plots as I showed you earlier, and we get about 120 nanomolar or something like that for the interaction of Wnt3 and frizzled 1. But there's something else. We see these interactions, these correlation functions only in this area, the midbrain hindbrain boundary and the roof lines. Sorry, the dorsal cerebellum. So there's only certain areas in which we see these interactions. Now, that was a question as to why would that be the case? And there was a nice article by Christoph Winkler's group that has shown that actually LRP5, which is a co-receptor for frizzled, is just expressed in these areas. So what we've done then, we knocked down LRP5 with morpholino, right? And looked again at the midbrain hindbrain boundary. And what you see is that that is what we had before. We see in these areas clearly interactions. So we see positive FCCS. The moment we knock down LRP5, there is no interaction anymore. So what that means is we need LRP5 together with frizzled to bind Wnt3. And now, because we know that now, we can test how much the binding of Wnt3 to its receptor, frizzled with the help of LRP5, changes the diffusion coefficient. So we have done the experiments with FRAP again, with Wnt3 EGFP, before and after LRP5 knockdown. And again, we get a recovery, and FRAP before knockdown is 0.5 micrometers squared per second. After we knock down LRP5, we get about 2.8 micrometers squared per second. So what that means, the binding to the receptor is another factor 5. So we have now a factor 4 times 5 times 5. There are about 2 orders of magnitude difference. We explained now, we see between FCS and FRAP. With FCS, because we are basically looking at a single spot within the extracellular space, we see the fast diffusion. But the moment we look at diffusion over a large area, we get tortuosity, geometrical effects, we get binding to HSPG, and we get binding to the receptor. And that together is sufficient to explain the difference between FCS and FRAP. So, short summary. So what we have explained now is the different interactions of Wnt3, with the receptor, the diffusion of Wnt3 in the extracellular space, and the interaction of Wnt3 with HSPG. Okay, so the last thing that is left over is the lipidation. So let's take a look at that very quickly. How many lipids does Wnt3 actually have? So for that, we used actually, again, imaging fluorescence correlation spectroscopy. So we use actually an array detector camera, but in this case, we use a light sheet. So we use actually one objective to create a light sheet, and with which we can actually eliminate any kind of cross-section in the sample. And under 90 degrees, we collect the data and put this on the camera. So we get at every single pixel, again, a correlation function of whatever we eliminate in our fish. Okay, now to explain this a little bit more in detail, we do now on the camera something which we call the FCS diffusion law. That's a particular analysis where we measure the time it takes to go through our observation area of a pixel, right? Independence of how large the pixel area is, right? If you get larger pixels, we have a larger observation area, the particle should take a longer time. Now, if you have free diffusion, what that just means is that the larger the area is, the larger is the time it takes for a particle to go through the area. And of course, that should go through zero, because if the area is zero, the particle takes no time to go through that. And that would be for free diffusion, right? For free diffusion, we know that the time it takes to go through an area is proportional to the area. What happens if you have something like trapping in domains? If you have trapping in domains, if you have very small areas, you get free diffusion. But if your areas are large enough that you start having actually that the likelihood of having domains in your observation area is high, then suddenly the molecules take a longer time to go through your observation area. Why? Because while they go through, they bind actually to some of these domains. So what you see then, you have free diffusion, and then suddenly you get more or less a kind of a jump, and then where the diffusion is now, or the time it takes to go through the areas is longer, and then afterwards it's just again average diffusion coefficient, which is now a bit slower because you bind to these domains. And Hop diffusion, which is not so interesting for us right now, but in Hop diffusion you have the same thing. If you measure actually the very small area, you get free diffusion. But if you go to larger areas, right, the molecule has to hop over these meshworks, and that doesn't happen all the time. So, but once it actually hops over it, it can diffuse further. But because it takes some time, it will slow down the particles. So what you see, if you're in the mesh, you have a very fast diffusion. If you're across the mesh, you get actually a slow diffusion. Now, the thing we can do, nothing of this we can actually measure here, because that's beyond the optical diffraction limit, right? So because diffraction is 200 nanometers, all these domains or these meshworks or the cytoskeleton or whatever is supposed to be in 50 nanometers or even smaller, 20 nanometers. So what we can do is we can extrapolate the data we get here to zero and take a look. Free diffusion gives you a zero intercept, y-intercept. Diffusion with trapping gives you a positive intercept. I hope you can see that. And a meshwork gives you a negative intercept. And what we're interested in is actually rather trapping, because your Wnt3 is lipidated with palmitoylation and myrstylation, which is probably more likely to go to some cholesterol-dependent domains or so. How do we do that in our case? Once you have a camera and you have recorded your data, afterwards you can decide what to do with the data. You can actually take one pixel and correlate it, but you can add up 4 pixels correlated, or you add up 9 pixels and correlated, right? So each one of them is a different area. So you get at least, alone from these 3 already, at least 3 different points. You can extrapolate to 0 and can say whether something is actually diffusing freely or not. So how did we do that? We took our zebrafish. We aim here at the cerebellum. We go to one of the cells, and go to the top of the cell. So we're at the top of the membrane with our light sheet, and then measure our correlation functions. The correlation function in the light sheet, the observation volume is a little bit larger because you have a slightly thicker light sheet, so they're a little bit more noisy, but they can be very well fit. And if we inhibit lipidation of Wnt3, we see actually that our diffusion coefficient goes up. So that's already a good sign, right? So if we remove some of the lipids from Wnt3, then okay, it moves faster. So now the question is, are there 1 or 2? Actually, there's only 2 sites which are supposed to be lipidated, and it's still not clear. Some people claim there's only 1 lipidation site. Some people say there are 2 lipidation sites, and this differs or might differ actually for different bins. So for Wnt3, what we've done now in experiments, we've done the following. So you have a control measurement, the round one, right? That gives you a clearly nice a positive intercept, meaning that Wnt3 sits on the membrane and it is in some kind of domains. If I put DMSO, or to put our fish in 1% DMSO, that's our control, because later on we're going to use the DMSO for the drug treatment, we get basically the same data. So here you see the diffusion coefficient, and that's and the blue is here on the right is the intercept. Here on the left, red on the left, diffusion coefficient, blue on the right is the intercept. Now we can add a drug, 2.5 micromolar of a drug called C59. C59 inhibits porcupine, which inhibits, because of the inhibition, is not lipidating Wnt3. So clearly we see a drop in the intercept. So Wnt3 is still on the membrane, despite us having actually removed some of the lipidation or inhibited some of the lipidation. It's still in the membrane, but it's not as trapped anymore as before. And if we put more of the C59 in, the lower the values go. Right, and that's what you see here. The diffusion coefficient actually goes up because there's less entrapment, and the intercept go down, right? Because it seems to get more and more in the direction of free diffusion. Just here a control. If we remove cholesterol with M-beta-CD in the fish, same thing, then we get a similar effect, right? So either we remove some of the lipidation of Wnt3 and it's less entrapped in domains, or we remove the domains by removing cholesterol and we get as well Wnt3, same thing. We get faster diffusion and lower diffusion loss. And then we just made a couple of controls for Lyn. For Lyn EGFP reacts to M-beta-CD, but it doesn't react to C59, which it shouldn't. C59 is only specific for porcupine and inhibits lipidation of Wnt3, but it doesn't do anything to Lyn, and that's exactly what you see. So C59 is doing nothing else to the membranes, right? Lyn doesn't care about C59, but Lyn cares about cholesterol. So if I remove the cholesterol, Lyn gets faster and the diffusion load drops. So that, for us at least, that clearly seems to indicate that Wnt3 has at least 2 lipidation sites. If you would have only 1 lipidation site and you inhibit it, then the Wnt3 wouldn't be on the membrane anymore. You wouldn't have a slow drop of the tau 0 and an increase of the diffusion coefficient. The diffusion coefficient would become very fast because now you have actually only soluble Wnt3 without lipids. But even when we inhibit at different levels the lipidation, Wnt3 is always on the membrane. That means we think at least there's only one of the lipids removed, and that makes it less likely to be in the domains, right? But it's still partially found in the domains. That's why this never drops completely to 0, and we still find the typical diffusion coefficient membranes, but it's not as slow as with 2 lipids, nor is it as much entrapped into membranes domains with 2 lipids. So with that, I'm not going to talk about the Wnt mutants. We have done a couple of Wnt mutants which show actually very similar data, because I think we're getting a little bit too long otherwise. So we have used combinations of FCS, FCCS, FRAP, and imaging FCS to investigate the dynamics of Wnt3 in live zebrafish embryo. I think that's actually for us a really nice thing, because you can really work in a physiological environment. The general discrepancy we saw actually between FCS and FRAP, right? This factor 100 difference or so. We can actually explain by tortuosity extracellular matrix proteins like HSPG and receptor binding. And we think Wnt3 is located in cholesterol-dependent domains. That's what the last measurements were about. And it contains 2 conserved lipidation sites, C80 and S212. I haven't mentioned that before, but these are the 2 sites which are typically expected to be lipidated. Sorry, yeah. And yeah, so that's, I think for us, it was a nice story, because you can see how you can use FCS in all kinds of different formats. You can use this as FCS, as FCCS, imaging FCS, right? And all this gives you different information within the embryo and can actually make up a whole nice story. In this case, we used FRAP as well to explain the discrepancy between short range diffusion and long range diffusion. And the main people who actually did the work was really Wen, who started actually a lot of these projects, Sapta, who finished most of it, and he's just left, and Indira was the I was doing actually all all the constructs. And I should mention as well, Ashwin actually, who helped with the Wnt3 mutants. Okay. Are there any questions?